Okay, based upon what we just learned in Mendel about the basics of genetic inheritance, so diploids, dominance, recessiveness, we can actually take this information and apply it pretty straightforwardly to human analysis. And so this is called pedigree analysis, and so this is typically used on rare genetic diseases. So how do we analyze natural populations resulting from uncontrolled crosses? So in this case, we do not have the control to determine who mates with who. So when it comes to model organisms, we decide which pea plant mates with which pea plant. But for human analysis, if we want to apply this to some sort of like medical situation, we don't have that control. And so pedigree analysis allows us to, one, determine the mode of inheritance of a disease. So is the disease dominant or recessive? And it also allows us to infer genotype information of individuals. So some of the basics of a pedigree. So this is a family tree with respect to a given trait. So this is an example, a couple examples right here of what a pedigree will look like. In order to interpret this, you just have to know a few things. So one, you need to know the sex of the individual. So the sex of the individual is indicated by shape. So female are indicated by circles, males are indicated by squares, and if the sex is unknown, then they're indicated by diamond shapes. If the individual has the trait or the disease, then they are shaded. So the affected individuals, you'll always notice, are shaded on this. Parents, so two individuals, they could be married or unmarried. If they have offspring, then that is indicated by a solid line, a solid horizontal line in between the two individuals. If those two individuals are related, so if they are cousins, for example, or if they are sisters or brothers, then that is further indicated by having these two horizontal lines together. So very often for these very rare diseases, having consanguineous parents where related individuals have offspring makes it much more likely that these rare alleles can combine in order to find a homozygous individual. So then these individuals have offspring, and so that is indicated by a vertical line that is attached to another generation of individuals. So this couple here had 4 different offspring. So they had a male son, the oldest first son was a male individual. They had 2 daughters that were younger, and then the 4th offspring was a male. So this tells us both the sex of the offspring and then their birth order as well. Fraternal twins, so dizygotic twins, so these are non-identical twins. So this vertical line will then bifurcate into 2 individuals. So this is how you can identify dizygotic twins. And then if they're identical twins, then you also have this extra line, this extra horizontal line right here. Sometimes if you have a large number of offspring and you don't want to indicate all of them on the pedigree, you can kind of simplify it. So if there are all multiple unaffected individuals, then you just include this number, and this would indicate that there's 4 daughters or 4 males. Proband, if you ever see this P within a pedigree, that is the proband. And so the proband is simply the individual that the doctor first saw that had the trait. So in this case, a male might have come to a doctor, described some sort of symptoms, and then the doctor decided to look to see, ask questions of this individual, and then the doctor might have asked, do other family members have this trait? And the proband individual might say yes, and so in that case, then the doctor would recommend getting a full pedigree. So the proband is simply the first person that kind of initiates the whole process of forming the pedigree. Deceased, if some individuals have died, this line right here is used. And then rarely you also see heterozygous carriers. So if you know that they're carriers, you can indicate that just simply using a dot in the middle. And then generations are also named in order. So what are some representative recessive and dominant human traits? So some recessive traits are albinism, color blindness is kind of a famous one, cystic fibrosis, that is pretty common as well, sickle cell anemia, so this is common from certain African populations, Tay-Sachs disease, and these are all recessive traits, so that means you have to be carriers of both of the recessive alleles in order to have one of these traits. There are also dominant traits, so Huntington's disease is one of the famous examples of that, and so in that case, you only need to be a carrier, and not a carrier, sorry, you only need to have one of the alleles and you will end up having Huntington disease. So this is simply the difference between a recessive trait and a dominant trait. That's kind of what this indicates. So how do you recognize that? So this is an example of a pedigree that you could use to recognize an autosomal recessive trait. So autosomal recessive traits typically skip generations. So if this is a recessive trait, then it's possible for it to skip a generation. Whereas in a dominant trait, it only appears in each generation. So the only way that you can have an offspring that has the trait is if one of the parents have the trait. So, you know, if this was a dominant trait, for example, why is this impossible? So this is impossible because if this was dominant, then we knew these two individuals would not have it. And so there would be no way for this individual to have any sort of genotype besides the little a. So you see, we just kind of figured out this individual can only pass on a little a, this individual can only pass on a little a. So there's no way if this was dominant that this individual would have it. And so this is what we referred to as a way to conclusively show that this is a recessive trait and not a dominant trait because of the skipping in generations. Let me see if I can erase this really quick. Okay. All right, and then in a dominant trait, so they almost always appear in each generation. Affected individuals also have an affected trait, and dominant autosomal traits appear equally in both sexes. So we will get to sex-linked traits later, but for now, we're just considering genes that fall on one of the autosomes. So this is an example of an autosomal dominant trait, and let's see. So, you know, you look at this and you notice that each of these individuals has it. Each generation has it. And then you'll also notice as soon as, for example, an individual loses it, then they are no longer able to pass on that trait. All right, so now that we know that this is a recessive trait, now we can also start to infer what the genotype are of different individuals. So because they have the trait, then we know that all three of these individuals must be little a. And so we can begin to fill in this as well. So the other thing is that if they don't have the trait, then there's two options that they can be, big A, or big A, little a. And so all of these other individuals must be one of those two genotypes, and that's what you're trying to figure out. So for example, we can look at these two individuals, and we know that they are carriers. So we'll just use this little dot to indicate that they're carriers. And so if they're carriers, that indicates that they do not have the disease, but they could potentially pass it on. So a carrier genotype is the heterozygote. And so we know that this is the only way there could be offspring that have the trait, is because, you know, if you had big A crossed with big A, little a, then all of those individuals would either be this or this. So just as an exercise, go ahead and kind of prove that to yourself. So the only way that you can have offspring that are homozygous recessive is if you have this cross right here. And so that was the way I was able to actually put this in there. So we can also know for certain that these individuals are carriers. And so that's because this individual has to pass on that little a to all three of their offspring. It's the only allele that they can pass on. And so these three individuals we also know are carriers. Another common assumption that we make is that if there's no evidence otherwise, because these diseases are very rare and the alleles are not very common in the population, is that if there's no evidence otherwise, a person marrying into the family is assumed to be homozygous and to not be carriers. So we'd also fill in this individual right here. We know that one of these two parents have to be carriers because they were able to pass on the disease allele to this individual right here. So 2,4 is a child of 1,3 and 1,4. And so the only way they can be carriers is to look back in time and to know that one of these two are carriers. But based upon this evidence right here, we don't know which one is. So there's a 50% chance that this individual is and a 50% chance that this individual is. And kind of consistent with the fact that both of these are not carriers, we don't see that there's any sort of affected offspring in this family. So only one of these two parents is likely to be a carrier. Again, we cannot, with conclusive say, is this person a carrier or not? Each one of these individuals has approximately a 50% chance of being a carrier and a 50% chance of not being a carrier. And then if we look here, we don't know for certain if this individual is a carrier. So this cross right here, all 4 genotypes are possible. So when you have a cross between two carriers, there's a 1/4 possibility that it is and then 1/4 possibility of that. And so basically when we look at this, we don't know for certain what this individual genotype is, but we can start to assign probabilities. One of the things we don't know, so we would not say that this individual is 1/4, has a 1/4 chance of being this and a 1/2 chance of being heterozygote. And the reason is that we know for certain that this individual does not have the disease. And so you have to do this renormalization process, and we'll cover that more in the activity. These again are identical twins, which is designated right here. So autosomal traits, autosomal dominant traits. So in this case, you typically start with the assumption, unless you have proof otherwise, that someone that has it is just heterozygote. So again, these disease alleles are very rare. And so because of that assumption, it's much more common for someone that has a dominant trait to just to be heterozygote. But for dominant traits, you can basically fill in all of the unaffected individuals, and all of them are little a, because if they had the disease allele, then they would have the trait. And so this makes sense. So this individual is little a, and that's possible with these two genotypes. So if this individual was homozygote, it would not be possible for this individual to have this offspring. So this we know is big A, little a. Anyone marrying in this is little a, and so forth. And so you can kind of go through this whole process and figure out the genotype of pretty much everyone within this pedigree right here. So that's kind of the idea. You look at these pedigrees, you try to figure out, is it a recessive trait or is it a dominant trait? And then you also try to assign genotypes to the individuals. And if you can't assign genotypes, then at minimum you assign probabilities that they have a given genotype.